Okay. Okay, well, I'm going to embarrass you a little bit first as we start off. For those of you that don't know, Duncan has announced his retirement about a month ago or so, effective July 1st. So it's a big loss for our division, for our department. And this is, you know, his swan song biostat seminar of many seminars that he's given over time. So I thought I would just, for those of you who aren't as familiar with him, a quick rundown on who Duncan is. He started his academic career back in 1972 at McGill as a lecturer and was assistant professor there. I never knew this, visiting fellow in Western Australia for a year. And in 1984 joined USC as, at the time, one of only really 3 card-carrying faculty with biostats expertise. Became full professor in 1988, and then has been a full professor and leader ever since then. Timed with his sabbaticals, he was a visiting fellow at the MRC unit in Cambridge, INSERM in France, and at Mayo for a brief time in 2003, and he was named the Bernard Richter Chair in Preventive Medicine in 1999, which is a chair that he's held ever since. Incredibly distinguished publication record with about 300 journal articles, many invitations to write chapters, given his breadth of expertise and monographs, and as you probably know, 2 textbooks, one in statistical genetics and genetic epidemiology, and the other statistical methods in environmental epidemiology. With respect to our education and training program, not his education and training, but our programs, he really developed the biostat program in our PhD program, demanding and developing many of the rigorous courses that span both theory and applications, and, you know, in my mind, define what a biostats dissertation should be, dating back from the early trainees that he had, and it still continues today to have that kind of approach to mixing theory, applications, and bringing those together in a rigorous dissertation. He's developed 4 of our main courses, 518, 533, 534. Couldn't remember the number for environmental epi.
575.
Okay, 575. And then initiated the 610 PhD seminar and has mentored a total of 52 postdocs, PhD students, and MS students over that time. And then, you know, has also been a leader both locally to our department and our division, and has really seen and overseen the growth from the original 3 biostat faculty. We now have 26 biostat faculty in our division. He's been crucial and played a leading role in the recruitment of just about all of those faculty. Also has played key roles on several of our big USC-based studies and grants, including the Children's Health Study, the multi-ethnic cohort, our PO1 in statistical methods for genomics, and he was, along with Stan Azen, the instigator and initiator of the environmental genomics T32, which is now in its 13th year. And then nationally, he was the president of the International Genetic Epi Society back in '99 and 2000. And he's been on many scientific and advisory panels covering topics of radiation, formaldehyde, drinking water, EMFs, air pollution, the list goes on. So I'll turn it over to Duncan here, but I want to just you know, mention that in addition to all this amazing scholarship and his scientific contributions, he really, I think, set a tone of collegiality, collaboration, and rigor that continues today, and I think sets our division apart from other divisions and departments of biostats across the nation and probably across the world. So can't thank him enough for his leadership, and you know, in true form, he can't be allowed to go out quietly. He's been invited by NIEHS to give a keynote speech, and he's going to be doing a dry run of that keynote speech today. So I'll stop there and turn it over to Duncan.
Well, thank you. If that wasn't embarrassing enough.
That was the point.
So let's see. I'm going to go into presentation mode. Now, they've given me half an hour to get through all of this stuff. So what I'm going to try to do is just walk right through it and then come back at the end for you guys all to take pot shots, tell me what doesn't work, and so on. But in the interest of trying to get a sense as to whether I can get through this in half an hour, let's hold your questions until the end, if you don't mind. So as Jim explained, this is for a workshop that's going to happen next week. And I'm the first speaker at 7 a.m. Our time in this workshop on integrating multi-scale geospatial environmental data into large population health studies. And there are going to be two of us so-called keynotes. Myself talking about gene environment, and Josh Apt, who's going to talk about sort of more of the geospatial kinds of things. So what I'm going to try to cover is, I think it should be evident why we're interested in gene environment. That's why you're all here anyway. And hopefully that this will become clear over the course of the talk. I'm going to devote maybe half my time to talking about some examples involving candidate genes and candidate, established exposure factors. But then devote the last half of the talk to talking about high dimensional data, some of the methodological issues that they bring up, and how we might relate multi-omics examples. I was going to conclude with the big picture, but in retrospect, I decided I would begin with that. So, come on. This slide is organized around an air pollution model, but the basic principles would apply to more or less any environmental exposure. And we would begin with some sort of external spatial temporal exposure field for which we have various predictors like traffic and weather. We have various measurements, central site or more localized, various modeling tools, spatial temporal modeling, dispersion modeling, and so forth. But what we're really interested in is a person's long-term average personal exposure, about which we have, again, all kinds of additional things that I'm not going to say anything more about, either to characterize individuals' exposure in comparison to the field in which they're moving, or possibly even personal measurements. From there, it's these personal exposures that would influence some underlying disease process, like, let's say, inflammation, leading to some ultimate outcome like asthma. And we would have various predictors, genes in particular in the present context, but also other host factors, and various biomarkers of what's going on inside. So for the remainder of this talk, I'm going to focus on this whole little box up here, and let others talk about the sort of exposure modeling side of things. And I hope what you learn from this is that exposure is complex. It's multidimensional, time varying, dose dependent. We've got all these different kinds of measurements to get a handle on it. And that's the subject that Josh is going to talk about in the next talk, as well as many other speakers throughout the next 2 days. I also want to make the point that both exposure and genotypes are high dimensional and will call for some novel study designs and analyses. And perhaps most importantly, we need to be able to incorporate biological knowledge formally into our epidemiologic analysis of gene-environment interactions. And I'll have a number of examples about how we propose to go about doing that. I'm going to do that by walking through 3 case studies, the Children's Health Study, or CHS, the Women's Environmental Cancer and Radiation Epidemiology Study, or WECAR, and the FIGI, or Functionally Informed Gene-Environment Interactions Study. And each of these illustrates a number of different principles which we'll get into in the next couple of slides. So let's start with the Children's Health Study. This is our cohort study of the long-term effects of air pollution in Southern California school kids. It entails about 15 communities with a wide range of air pollution, and they've been followed since about 1990. This study was originally conceived around a hypothesis that was elaborated by Frank Gilliland involving oxidative stress, as well as the various determinants thereof, xenobiotic metabolism, inflammation, and so forth, and the various modifying factors that lead to health effects. So for this purpose, we had a complex system of exposure assessment involving measurements of ambient exposures, traffic density, and so on, that people will speak about, including Rima Habri, who's coming up later today. We also focused on a variety of candidate genes inspired by Frank Gilliland's picture. And this picture simply describes those various genes as they're involved in various stages in the process of creating oxidative radicals and their detoxification. So one of the first things that we learned in terms of gene-environment interactions was that maternal smoking interacted with parental history of asthma, such that it really seemed to have no effect in people that did not have a parental history of asthma, but had a strong and, more importantly, an interactive effect in those that did. So amongst the genes up here that are potentially relevant to this process are the glutathionase transferases, in particular GSTM1, that we showed had a multiplicative interaction, a greater than multiplicative interaction with maternal smoking while pregnant, but not after the child was born. We then went on to conduct a GWAS. We've done numerous studies of epigenetics. We've talked about metabolomics. These are all things that I will come back to in the second half of the talk. But before leaving this study, I wanted to talk about our thoughts about incorporating functional data. So here we have a sort of a schematic of the CHS involving measurements of ambient pollution, individuals' genotypes leading to various health outcomes. And what Rob McConnell and David Diaz-Sanchez did was take samples from the filters that we were recording ambient pollution on and dust them onto cell lines constructed from some of our individual subjects, so representing a spectrum of genotypes. And then they measured a variety of different biological activities. So our goal here was to formally incorporate this toxicological information into our analysis of the epidemiologic data, either by simply taking predicted biological activity as a risk factor in the epi analyses, or by relating the coefficients of the prediction model for biological activity to the regression coefficients for the gene-environment interactions. WECAR is next. This is a study of second breast cancers in relation to radiotherapy and the various DNA repair genes that might be involved. And it too was organized around an a priori hypothesis outlined by Pat Cannon that entails DNA double-strand breaks being caused by ionizing radiation, those breaks being recognized by the ATM gene, which in turn phosphorylates a whole series of other genes that are involved in various damage response pathways, DNA repair pathways like non-homologous end joining or homologous recombination, as well as apoptosis, checkpoint control, and so forth. We conducted a nested case control study in 2 phases, the first involving 700 contralateral breast cancers, second breast cancers in the non-irradiated breast, and 1,400 unilateral breast cancers who'd survived to the same age without getting a second cancer. And then for replication, we added another 800 plus 800. The first of these 2 stages was designed with a counter-matching approach that I won't talk about in detail, but suffice it to say that this technique introduced by Brian Langholz considerably improves the power of both for detecting the main effect of radiation as well as radiation gene interactions. Phantom dosimetry was conducted by Marilyn Stovall at the MD Anderson. And this schematic basically shows the exposed breast here getting about 4.5 to 6, 45 to 6 gray of radiation, and the scatter radiation to the contralateral breast, which is the subject that we're interested in. With these data, she was able to show a dose-response relationship with that was stronger in those subgroups where we would expect it, based on things like the atomic bomb survivors data and other radiation cohorts. So after 5 or more years of latency, a stronger relative risk, stronger in young women, stronger yet in those with both. In terms of candidate genes, obviously the first candidate of primary interest to us was the ATM gene. We sequenced that gene, found 242 unique variants in that gene. And we were able to classify them using the SIFT software, sorting intolerant from tolerant. And amongst those that were classified by SIFT as likely deleterious, we saw an elevated relative risk associated with those variants. But more importantly, it was those variants that showed a strong interactive effect with the radiation exposure. And again, that radiation effect was strongest in those who were exposed as young women. Now, you might wonder about BRCA. That is, as well as being a strong risk factor for first breast cancer, also a strong risk factor for second breast cancers. But when we looked at for radiation interactions, we found no multiplicative interaction. But this begs the question of what do you tell a woman who's carrying one of these mutations about whether she should get radiotherapy? Given that she's already at increased risk, do you want to subject her to any further increase? And this is something we argued about considerably, given that the interaction effect was not significant, although greater than additive. Again, we did a GWAS, which I'll tell you more about later. But we also talked about integrating functional data. And for this purpose, what we thought we would do is take a random sample of 5 of those individuals that had the most common mutations, and all of those that had rarer mutations, constructed cell lines for each of those, and assayed a whole bunch of different biological endpoints, including things like colony survival after radiation exposure. And the idea was to use those as prior covariates in the kinds of hierarchical modeling I'll talk about in the second half. Finally, we turn to the Fiji study. This is a consortium of 62 different studies totaling about 130 subjects, all of them with GWAS data that could be imputed to a common set of 30 million SNPs. Also, all of them with harmonized data on 14 established risk factors that you'll see in the next slide. And I will come back in the second half and talk about some of our approaches to genome-wide scanning for gene-environment interactions, and also incorporating external information. In my mind, one of the most exciting aspects of this whole study. So we began with constructing a genetic risk score and looked at its interactions with those 14 risk factors, environmental risk factors that I mentioned before, that show quite a range. Colorectal cancer is one of those cancers that has quite a rich range of different modifiable exposure factors. So the potential for targeting interventions is very good for this cancer. At the stage where we're doing these analyses, there were 27 GWAS hits, which we constructed into a genetic risk score that showed a roughly twofold range of variation over the interquartile range of genetic risk scores. And then when we broke it down by these 14 risk factors, really nothing except for amongst women, non-use of hormone replacement therapy showed any departure from a simple multiplicative model. More departures in terms of additive interactions, although at least some of these things like height, we would not exactly consider a modifiable risk factor. But then things like processed meats also emerged as a greater than additive, but not greater than multiplicative interaction. So with data like this, we wanted to ask the question, should we target screening to those that are at highest risk? And Li Su's first paper on this subject addressed this using that same risk score that I described previously, and computed the target age at which screening should begin as a function of gender, the red and blue lines, whether or not they have a positive or a negative family history, and the distribution of that genetic risk score. So that women with a low genetic risk score and no family history might safely wait until maybe age 57 to start screening, whereas men with a positive family history and a high genetic risk score should probably start in the low 40s. Now, there's no environmental risk factors in this particular analysis, but we followed that up later with an analysis of an environmental risk score, much like the genetic risk score, here restricted to those that are modifiable, so like not height, and now found a much broader range of starting ages at screening, ranging from the late 60s for the low risk factor family history negative females to about 40 for the high risk individuals. Now, as I said, one of the most exciting aspects of this study is the incorporation of functional data, for which we have two sources. First is the so-called Barsuva UVA sequencing study, which entails 445 normal colon biopsies on which we've measured gene expression and have information on their past exposures and their genotypes. We also have constructed mini-guts, organoids constructed from stem cells from normal colon crypts, on whom we again have measured expression. And rather than the observational exposures that we have up here, we have experimentally applied exposures to things like alcohol, folate, and so on, as well as the genotypes for those colon cancers. So for each of those various exposure factors, we're able to evaluate exposure-specific gene expression and incorporate that into our epidemiologic analysis. And I'll come back to tell more about that now as we get into the second half of the talk. So there's two main things I'm going to try to do here. One, simply talk about some of our approaches to high dimensional data, and then illustrate that with a range of omics types and even talk about multi-omics. So the first of these approaches, I want to talk about Bayesian hierarchical models, which are one set of machinery for incorporating external knowledge about pathways or other kinds of knowledge into an epidemiologic analysis. So what makes it a hierarchical model would be the first level model would be perhaps a fairly conventional logistic regression model involving main effects and interactions, each of them with a regression coefficient. And then because the data are high dimensional, often with many more terms in this model than we have subjects, they need to be regularized in some fashion, such as using the ridge or a lasso prior. And this slide simply illustrates doing that on the 12 most common ATM variants, showing the unconstrained estimates. Here there was only 12, so we can actually get maximum likelihood estimates, but with large variability around them, and the results of after penalization. Smaller estimates shrunken towards zero, but also with tighter confidence bands. But I contend that we can do better by taking those regression coefficients down to a second level regression, where we would regress them on various pieces of prior knowledge, such as those that I've described in the previous examples. So just one analysis of that. This is an analysis of WECAR data, including not just the ATM, but 39 other candidate DNA damage response genes, and then using their various Go terms from the gene ontology as prior covariates, showing that things like apoptosis and especially DNA repair via the homologous recombination repair pathway are associated with larger effects. Now, what about genome-wide interaction studies, or GWIS, as I like to call them? In 2008, Cooperberg and LeBlanc from the Hutch described a two-step approach that begins with an initial scan of associations between the gene and the disease marginally, and then taking the top ranked of those SNPs and restricting the search for gene-environment interactions to those that passed some threshold, and they showed that you could get considerably improved power for that method. Our own PhD student, Cassandra McCrae, along with Jim Gauderman, adopted a similar approach by testing instead for the association between exposures and genes in that first stage, much in the spirit of a case-only analysis, but here using the combined case and control data set so as to get a test which would be independent of the test for the second stage interactions. And they showed that that also improved power. And then went on to develop a two-step approach they called EDGE, which combines the exposure gene and the disease gene screens into a 2-degree of freedom first step test, and then restrict the test for gene by environment interactions to that second step. And they showed that there is no one universally most powerful method, that it depends upon things like the marginal gene effect, but that over a range of effect sizes for this and other variables, the EDGE approach does quite well compared to many of the competing methods. All this has been absorbed, has been included in a piece of software called GxE Scan. It's an R package that's been downloaded numerous times and is just helpful for study design, for actually conducting these scans and outputting a lot of the kinds of graphics that one would like to have for the various papers. So in terms of applications to the Fiji data, I'm going to show you some very preliminary results by Christina Jordal, a postdoc at the Hutch, still unpublished, but the first of what will be a whole series of papers describing gene-environment interactions with various established risk factors in the Fiji study. This slide just shows a overall test of an exhaustive scan for all possible gene-environment interactions with the 30 million SNPs. None of them attain genome-wide significance, but the blue line here represents what we're calling suggestive significance, and includes a couple that involved already established genotypes in the chromosome 10 region. This describes our two-step weighted hypothesis approach introduced by Ionita Lazo, and now the orange dots here represents a number of those from that previous scan that now attain the threshold for genome-wide significance in this two-step approach. So one of those, this particular genotype, shows an interaction with moderate alcohol consumption, such that the protective effect of moderate alcohol compared to none, or compared to heavy, which is an adverse risk factor, that interaction appears only in this category and not in the other two. So zooming in on that region, there are a number of SNPs that have been that form that pile that you saw before, and they tend to be clustered in the region of two genes, ENTPD7 and COX-15. So by constructing a standardized a predicted expression score, it by itself marginally has no predictive value for colorectal cancer. These are the same marginal effects of alcohol consumption, showing the protective effect of moderate and the deleterious effect of heavy. Whether this protective effect is a real biological effect or due to something weird about people who don't drink alcohol remains to be seen. But again, the point is that that expression variable interacts with moderate alcohol consumption, but not with heavy. Now I want to turn to some other kinds of omics. Again, sticking with the Fiji study, we have a study of the organoids that are exposed to either alcohol or control that identified a whole slew of differentially expressed genes, which could then be related to various pathways. And the authors concluded then that they saw an enrichment of pathways for genes related to ethanol metabolism and or colon cancer. So what could we do with data like this? Well, the commonly used technique would be to filter the associations so that we focus attention just on those that are supported by biological information. And that, in a sense, is a form of taking all or nothing weights. We can also use them in that two-step approach, where we would create a score that's based on that biological information, and then use that to decide which genes to include in the second step. Or we use it as prior covariates in a hierarchical model. Now turning to some other epigenetics, other omics approaches, my children's cell study colleagues came up with this picture to illustrate the effect of the arginine and nitric oxide pathways. It's a complicated picture that I won't talk you through in detail, but the point is that it was the motivation for a whole series of subsequent papers that came from the children's health study that I'm just going to illustrate very briefly diagrammatically. So one of the first of these is a paper by Mohammed Salam that focused on the two genes, the ARG and the NOS genes, and showing an interaction with ambient ozone in the production of childhood asthma. Another takes those same genes and looks at exhaled nitric oxide, a measure of systemic inflammation, and showed how that was modified by the presence of asthma. Another looked at the methylation of the ARG gene and its modification by asthma. Another showed a three-way interaction between the NOS gene, NOS methylation, and particulate pollution. Others looked at NOS and lung function in relation to asthma. You put it all together, we get this very complicated picture, which I tried to simplify in terms of statistical data set that involves the ARG and NOS genotypes, the ARG and NOS methylation, particulate pollution. I didn't have expressions, so those become latent variables in this model, leading to excelled NO with asthma as a modifying factor. And with that, I was able to show that it was in principle possible to estimate those various effects. Now, what about multi-generational effects? My former colleague Brian Langholz, together with his student Yufeng Li, demonstrated that, both maternal and grandmaternal smoking while pregnant produced an increasing effect in the production of childhood asthma. Carrie Breton then and colleagues went on to look at the methylation of various genes that are potentially involved in that process and showed a number of those that were significantly associated with prenatal with prenatal smoking, as well as with global methylation, line 1 and alu methylation. So putting these various things together, I came up with a picture for how one might want to go about doing multi-generational inheritance. And sorry about that. I thought I persuaded him to blow later. Anyway, so taking data on grandparental genotypes and exposures, one might construct a latent variable for grandparental epigenotypes, which could be transmitted to the next generation, interacted with their exposures, and on down to the offspring generation, interacting with their exposures. So that's not an analysis we've actually conducted until now. And Carrie Breton, together with her student, Sarah, are working on just such an analysis right now to relate prenatal smoking of the grandmother to the offspring's wheeze and conduct a causal mediation analysis involving DNA methylation in both the maternal and the offspring's genotype, as well as mediation through the parental and offspring's wheeze. Finally, I want to talk a little bit about metabolomics and microbiome. This is work being done by my colleague Zhanghua Chen, using a sub-study of 102 asthmatics from the Children's Health Study. XMWAS, a standard metabolomics analysis package, constructed this network that relates air pollution, non-freeway NOx, PM, and so on, to expression of 357 genes. Those are the triangles on the outside here, and 92 metabolites. These are untargeted metabolites, the various circles here, and there are correlations amongst them. Using another package known as MetaboAnalyst, they were then able to identify various pathways that are involved. PM 2.5 and NO2 were primarily associated with amino acid metabolism. Non-freeway NOx with oxidative phosphorylation, ozone with, sorry, I can't see my own slide there, and so on. And then putting it all together, they come up with this elegant picture for how it all fits together. In terms of the microbiome, again, in another sub-study of the Children's Health Study, 16S sequencing led to the identification of two families of bacteria, one of which is negatively associated with freeway NOx, the other positively associated, and showed that fasting glucose was associated with those same, the relative abundance of those same families of bacteria in the same direction. So what about tying them all together in some sort of multi-omic fashion? One approach, using this MMVEC approach, clusters the metabolites and the metabolites and the microbes based on their correlations across subjects and reveals a number of different categories, which then we could look at in relation to exposure and see that both the microbial clusters and especially the metabolite clusters strongly related to ozone exposure. Another way of doing it is to cluster the subjects based on the similarity of their profiles of metabolites and microbes. And again, we could see that that clustering into now 3 different categories of subjects also strongly related to ozone. So with that, I'm going to stop. Thanks to many, many people that have contributed to this. This is a hardly complete list, but I want to thank, in particular, the various key investigators from the CHS, WECAR, and Fiji, as well as my own colleagues on our statistical methodology program project grant. Thank you. And with that, I will quit. Okay, guys, pot shots time. Tell me what worked, what didn't work. I will review the timings later, but I suspect that was more than half an hour.
Yeah, it was 35, right on.
Okay.
You're close.
I'm close. Every time I practice for myself, of course, it was 30 minutes, but I tend to ramble when I've got an audience.
Yeah.
So what should I chop if I have to chop?
It's an amazing collection. I mean, it's really spans a lot of interesting study designs and all of that. I found the last couple slides, the MWAS and the one of the other ones.
Analyst.
Yeah, it got a little hard to follow. I think you were rushed for time as well. So there was a lot of material on those slides, and I'm not sure the message that you might have wanted to get across quite got across. Then I saw you kind of tied it together later on when you were bringing the metabolomics and the microbiome together, I think. So, you know, I'm not sure if you can cut any of the ones just before that, or if you need those to kind of round out that story.
Well, you're quite right that that's material that I'm probably the least comfortable with of what I'm showing there. And in general, I feel like in my attempt to try to be comprehensive here, I'm being somewhat cursory in my covering of a lot of this.
Yeah, I mean, you have a lot of material, so I think nobody will miss it if you don't have it. But if you need some of that material to make one of the key points, you might be able to reorganize that just a little bit. I'll let others chime in.
Jim, I would like to ask a question. First of all, thanks to Jim for providing the opening plaudits for Professor Thomas. I will say that I've been a fan of his work for decades, and we will not also escape the opportunity to embarrass you further with some sort of reception because your retirement will certainly not go unnoticed for the department. But I do have a question. As an environmental epidemiologist, I noticed at the very beginning, you put up a sort of model of how you looked at the universe of elements that go into thinking about disease. But exposure was sort of characterized as sort of some sort of integrated measure of exposure. And I guess for those of us who are doing all these kind of life course cohort studies, exposure gets very complicated. It's not just cumulative exposure. We have to wonder whether there's peak exposures that might be most important for disease, or perhaps exposures at different life stage. And I guess my question is, you know, do these various models accommodate a approach where you're agnostic about what what metric of exposure might be most important and use that to actually identify which exposure metric might be the most important in GE interactions.
Indeed, that's certainly true. And that's why on the slide that follows, my take-home message is, I point out the time-dependent nature of exposures. And I chose not to really elaborate on that just because that's what many other people in this workshop are going to be talking about. And I wanted to focus on the genetic side. But yes, indeed, that has been the whole, exposure time response relationships has been a long-term interest of mine. So I can only conclude from the silence there that nobody wants to take pot shots, that you didn't understand anything. And so if I were to eliminate basically all of the slides, you'd be happy.
Not that at all. So what's the audience for this?
Good question. Let me show you the program. Give me 1 sec to call it up here. Okay, I've found it. Now I just need to get back to Zoom. I can find where Zoom went. So I can share my screen again. Okay, you see this?
Yeah.
So now somewhere in here I can get agenda. There we go. So it is really heavy on the exposure side of things, more than the genetic side. After Josh App's talk, there will be a whole session on geospatial technologies. So that's where Rima is talking. And so these are things like satellite monitoring and personal exposures and all of this kind of stuff. Then more spatial temporal modeling, measurement error, and this sort of thing. So then on day 2, big data. So here's where we might expect there to be more omics. And other than this one talk here, which is also geospatial in nature, the other 2 talks are, again, still mainly high dimensional exposure data. And then a final presentation on tying it all together. So I think that it's going to be primarily environmental epidemiologists that will be there.
I guess I was wondering, how much are they going to be familiar with, what gene expression data looks like, what metabolomic data, microbiome, do they need a little bit of, a primer on that to follow some of the things, or do you think they should be pretty up on that?
Well, that's a good question. I'm loath to do that just because of time constraints.
Yeah.
What I sort of want to leave them with the message is that, I'm sorry, I forgot that there was this another whole session here that Rima is I guess, chairing, that involves things like GxE for toxicology and so on. But anyway, I wanted to leave them with the message that neither exposure nor genes nor the tying of them together is simple, that there's a lot of potential for using high dimensional data, and that it's not just one gene and one environment, but a whole host of them and a whole host of biomarkers that we want to use.
Yeah.
So I hope that message comes across.
Yeah.
But you're right that I haven't really thought enough about my audience and whether I'm going to be bringing them along with me, those of them that are not particularly involved in genetics.
Yeah, I don't think you have to do much. I mean, I think we can probably assume they know generally the kinds of data, but you know, I was thinking about the expression results you showed for alcohol, which you know, I think there's an opportunity there to maybe take 15 seconds and just, you know, remind them that the predicted expression comes from that Barsuva data, and that we predict that back into the bigger sample. So it's kind of a, you know, that's where we're at with some of the omics, is that we have smaller sample sizes and we have to build prediction models, and then try to use those back on the larger resource to make inferences. So you would spend a little while introducing the Barsuva data. It just might be worth re-mentioning it here as an example.
Good suggestion. And I think I can do that with this, adding a footnote here, which it's not letting me do it right now, but.
Or you can just say it, you know,
The footnote is not more for my own purposes to remind me to say it.
Yeah.
Anyway, I will stop fussing with it. This will be enough to remind me to do it.
Yeah. But I mean, you know, you cover generally a nice waterfront, you know, lots of different technologies and levels of data availability. And, you know, I think it's going to really set the stage for the 2-day meeting probably. And you'll probably have lots of side conversations afterwards for people.
Yeah, that is the point.
Yeah.
Other suggestions? Let me just very quickly step through them and stop me if it reminds you of anything here. I thought about actually ditching my initial outline and jumping right into it. Not sure whether I want to do that or not. I've been gone over and over it, back and forth. It would save me about a minute.
Yeah. Could you do that intro in the context of your big picture, do you think? Rather than spend time on this slide.
I sort of can. I can say it here. But there's nothing particularly high dimensional in here unless I say it.
Yeah.
Let me think on that some more. There are a number of similar signposting slides scattered throughout the talk.
Yeah.
So this one, then my 3 examples.
Yeah.
I think I spent about 2 minutes roughly on the CHS.
I think that will be of interest to this group for sure.
For sure, yeah. Now this one, because I wanted to have an example from each one of these you will recall is a study that actually got funded, but we never got the data because David moved. But I don't want to give him a hard time about that. So I'm just going to imply that that this is something that we really thought about doing.
Mm-hmm. Mm-hmm.
But I'm not showing any results from this one. Likewise, for the WECAR study, I spent probably about 2 minutes going through this stuff. I may decide to drop the BRCA piece as not essential and focus just on the ATM part of the story.
You could.
But for me, it represents an important piece of clinical implications.
Yeah.
So again, this is a study which we proposed and never got funded. So I still wish we could, because I think it would be really interesting. But again, I have no results to show here. So then we get to Fiji. I spent probably the longest time on this because we have the most to talk about. Even though this never got published because it got superseded eventually by the E-score paper.
Yeah.
I still like that presentation. And then again, this sort of talks about targeted prevention and the incorporation of environment into that. And then this is the important slide.
Yeah.
So with that, I then detour into methods briefly. I'm kind of sick of talking about hierarchical models because I've been doing it for so many years now, but and this is maybe not the best example I could come up with, but I might go back and take a look at David's BRCA variant paper, see if that makes a better example. And then in terms of the pathways, what I would like to do is really add something about how that has changed the ranking of the various polymorphisms. So let me think about that. Oops, wrong direction. So then we get to GWIZ. Did I make the right noises about GxE scan?
Yeah, that was, and you got through that really quick, so I think that's good.
Yeah. And then this one. So as you know, this is one that we've been struggling with a lot over the last few weeks.
Right.
But I'm pretty happy with it now.
Yeah, you know, if you want to save a little bit of time, you could maybe even omit the upper left figure and just say the two-step method found SNPs in this locus, and then point to the locus zoom and that it's in those two genes.
I could, though that's my only slide on the two.
Yeah. I mean, I'm loath to have you get rid of something that, you know, our group generated, but yeah.
Plus the original scan is totally non-significant.
Yeah, that's a good point. Yeah.
That's why I included it.
Yeah, okay, keep it.
I don't really have very much to say about this. What you can't see right now is the path, this bit. And I couldn't think of anything intelligent to say about the top ranked pathways here. So instead, I adopted the quote from the paper itself.
Yeah.
I think that works.
Yeah, that was fine.
Whoops.
That figure, though, is hard to understand unless, well, at least let me say, I found that figure hard to understand.
So hard because you can't read it, or because
probably a bit. It's certainly, that's a prerequisite. I can't read it, so. But I suspect even if I could, I would need someone to explain what the XEs were and how I should interpret that figure, not knowing this stuff very well.
Okay.
I think I mean, I don't know if you need this slide. You mentioned that Graham had done those experiments earlier.
Yeah.
I know this is the only result coming out of that, but.
Well, again, it's alcohol, so it ties in with the previous slide.
Right.
And I don't particularly like either of the two graphics there. Previously, I have a version that I omitted here, where I highlighted the top ranked one and tried to explain why that was maybe interesting, but I'm making it up. I have no idea really whether this has anything to do with either alcohol. As far as I could tell, this gene has nothing to do with alcohol, and whether or not TGF-beta is particularly important in colorectal cancer. It seems to be important in just about everything anybody looks at, but I don't know. I'm out of my depth there, and I'm not sure that I should be trying to explain what some of these top-ranked genes are.
Yeah.
So I decided to ditch that. So anyway, then we get to this whole series, which I illustrate just to show how complicated these pathways are. And I don't know if that works at all. Did you guys like what I did with this? Or is it just too much too fast?
I like this. I think your audience will like this because it gets back to, you know, a respiratory outcome and air pollution.
Yep.
I think this is a good one. I actually like this part a lot.
Okay, good. Well, I'm talking to a statistician now, so.
But I mean, I think the point here is that you've brought together exposures, intermediate variables, you know, outcomes, et cetera, and that, you know, there are ways to try to tie all this together. I think that's the important bit of this. And there's some, you know, intermediate variables still out there that, you know, some may be measurable now that weren't measurable in the past.
Exactly. A little nervous about getting into multi-generational effects, given how controversial they are. And my main problem with this slide is I don't know whether these genes, how these genes were selected and whether they have any relevance to either tobacco smoking or our outcome variables.
Yeah.
They were the ones that were popped up as being methylated differentially with respect to prenatal tobacco smoke.
Yeah, I wrote a little note that I wasn't sure you needed this or the next slide, which I think is related, right?
Yeah, this one I'm inclined to ditch. I spent more time over it than I have in my practice sessions. And I'm inclined to get rid of that, but maybe keep this one.
Yeah, I think if you had, I mean, my sense was if you had to cut one little unit, slides 39 to 41 maybe could be cut, unless there again, if it's leading into something you're going to say a few slides later, then I guess, you know, you may need to keep it. But this went by awfully quickly and it was a bit hard to follow for me at least.
I think I will ditch this one.
Okay.
And I'll decide about the other two.
Yeah, okay.
And then finally we get to the ones that you had a hard time understanding. And that's largely my fault, not doing a good job of explaining them. But I will work with Zhang Hua. She did a beautiful job of walking us through her version of these next 4 slides during my 575 class this spring.
Yeah.
So if only I had recorded it. Well, actually, come to think of it, they are recorded on Blackboard.
Oh.
So maybe I'll go back and see what she actually said about them. That one's fairly self-explanatory, and these I think did okay.
Yeah.
And then an abrupt conclusion, because I took out the slide that I had about future challenges and decided I was out of time. So I won't try to summarize.
Yeah.
But maybe I should. I could just bring back my take-home messages.
Yeah.
And then I don't need to talk about it because I already have.
Right. That's a good idea. Yep.
Well, it's already past 1 o'clock. Anybody have any further suggestions?
You need to add Paul Marjoram to that last slide.
Oh, sorry, Paul.
That's all right. I didn't even notice I was not on it. So.
Oh, what do you know?
Or that I should have been.
Yeah. Done.
And I think you got everybody else.
Thank you for your patience to listen to me, and wish me luck on Tuesday.
Yeah.
Good luck.
Yeah, luck. So I'll be interested to hear how it goes.
Yeah.
Yeah. Will you be back for trying to figure out the image schedule since it's just the few of us left here? Kim had proposed that we cancel until after July 4th, but I've been going over the budgets for year 6 with Chris and Nicole, and it might be worth us having a huddle. At least the PIs of each project and core maybe could huddle at next Thursday's planned meeting. And Duncan, if you're back and you want to do it, it'd be great to have you join as well. But
Yeah, I will be available.
okay.
Okay, just to clarify, this is not a virtual conference. This is going to be in person. Is that right?
No, it's virtual.
Oh, it is. Oh, yeah. Okay. Yeah. Okay. Well, I was hopeful you were going to get to travel, but I guess not.
Well, those dates will still be a little while in the future.
Yeah. All right.
Okay, well, thank you all.
You bet. You bet.
Good luck.
Good job.